All right. So this warm up was all about standard 1.1.3, which is cell differentiation, aka cell specialization. We discussed this concept yesterday, and it is such an important concept for us to get. We've really got to tie in this whole idea of protein synthesis now that we've talked about transcription, translation, gene regulation, and cell differentiation. It's kind of that linear process that takes place to allow a cell to become specialized and to give us the traits that make us what we are, not just us, not just human beings, but all living organisms. Okay, so the first question asks, how do stem cells become specialized to absorb nutrients from food as intestinal cells? How do they become specialized? Is it by gene removal, by gene activation, through transcription, or through translation? Good, Janelle, and I appreciate that you typed it out instead of just saying it out loud. Gene activation is the correct answer. So we've talked about this yesterday, and it's gonna. You guys will probably get tired of hearing me say this over the next few minutes, but gene activation is the process by which a cell turns on a portion of its DNA, a very small portion, and the vast majority of the DNA is left deactivated. Different cells will activate different genes. In doing so, they produce different proteins, and in producing different proteins, they end up with different traits. Okay, so it's a super important linear process. I activate some genes, I produce a specific set of proteins, and that gives me a specific structure, which gives me a specific function. Other cells might do things differently. They do things differently. Question 2, how are the enzymes that nerve cells produce different from the enzymes that blood cells produce? Yes, Ashley? Was that an accident?
The genes are activated in nerve cells are different from those activated in the blood cells.
Good. Excellent. So again, we're activating different genes in different cell types. This is what gives them different qualities. Every cell has the exact same copy of DNA. They all have your whole genome, but they will activate different parts of that genome to give them different qualities. Third question, which statement about the genes of specialized cells is true? All the specialized cells of an organism contain the exact same DNA. Each specialized cell in an organism contains a different set of DNA. The specialized cells of an organism contain twice as many genes as unspecialized cells, or the specialized cells of an organism contain half as many genes as an unspecialized cell. What do we think? I kind of just said it just before. Good. So thank you, Melissa, and thank you, Janelle, who was talking behind me. I don't know if you all could hear that. All specialized cells in an organism contain the exact same DNA, the exact same DNA. And this is, if we think about it in this way, for those of us who are interested in criminology or forensic science or criminal pathology, this is how investigators are able to figure out who has been at a crime scene, regardless of what biological substance is left on the scene. It could be hair, it could be blood, it could be, in some cases, saliva, it could be skin, it could even be a fingernail. These things all can help to identify who was present at a crime scene because they all have the same DNA. They all have the same DNA. They just have different genes activated that give them different qualities. Your blood cells obviously are not the same as the cells that might be in your saliva. but they are coming from the same person, so they have the same DNA. Last question. Myosin is plentiful in muscle cells, but it is not found at all in lens cells. Lens cells contain large amounts of crystallins, which are not found at all in muscle cells. Why do some proteins appear in one type of cell, but not in another? So here's a good example of a test question where we've struggled on the first 4 tests of the class so far, because it gives you all this long information about things that we have not covered. We've never talked about myosin. We don't know what a lens cell is. We've never talked about crystalline. But those are all distractors, essentially. They're just giving you an example that might help to color your answer a little bit. But for the most part, we just need to focus on this part. Why do some proteins appear in one type of cell but not in another? That's the question. It didn't really even help us to have these 2 sentences. So we've got to get in the habit of being able to identify the most important part of the question, which is usually the question itself. So what do we think? Oh, we've already got an answer. Certain genes are activated to produce, yes, good. Excellent. So Ashley has dropped the answer in the chat. During differentiation, certain genes are activated to produce proteins that enable specific functions. Good. So I won't run through that linear relationship again, but it's such an important one. All right, let's jump back over here. I'm going to stay. Okay. I'm already recording. So today's March 3rd, 2021. Let's go ahead and start getting ready to take some good notes today. It's the 6th day of our 5th unit. And we're going to talk about mutations today. This is really the last bit of new information we're introducing in this unit. So tomorrow we will review, Friday we will review, Monday we will review, and then Tuesday you all will take your midterm. And then for the rest of next week, we will likely be doing some fun activities, but also giving those of you an opportunity to, those of you who need it, an opportunity to catch up and get some missing assignments turned in. Keep in mind that the quarter ends next Friday. Next Friday is also a teacher work day, so you all will not have class, which means we really need to get everything done by next Thursday. Don't wait until then, though, because if you have missing assignments, the best way to prepare for your midterm, which I will remind you is about 20% of your overall grade, the best way to prepare for that is just to knock out those assignments. Okay. So the objective today, biologists will be able to explain how mutations in DNA that result from interactions with the environment or new combinations in existing genes lead to changes in function and phenotype. That's a mouthful. What we really want to think about is this word mutations. I'm sure we already have some ideas about what that means. We've heard it before. And then we want to kind of think about where these mutations are coming from and what do they do? So the essential question, how do mistakes in the process of protein synthesis affect living organisms? What effect do these mutations have on living organisms? All right, we cool? So right off the bat, a couple of definitions for you to write down. First one is mutation. Mutation is defined as a change in the nucleotide sequence of an organism's DNA, or in the DNA or RNA of a virus. I didn't highlight that because we won't talk much about viruses in this class, but it's good to know that viruses are often caused by mutations, and then they benefit from those mutations. For example, you're hearing about all these different strains of coronavirus, the South African strain, the Italian strain, the Israeli strain, the California strain. There are different strains because, yes, they're all the same virus, the coronavirus. the novel coronavirus. But they have slight mutations that give them different properties. Interestingly enough, typically viruses become less deadly over time because the virus doesn't want to actually kill off its host, at least not immediately. A virus wants the host to continue to live so that it can produce even more virus. So it's actually kind of counterproductive if the virus is so deadly that the person, its host, is not actually able to produce more virus. So over time, because of natural selection, viruses pick up mutations that make them less deadly, but also that allow them to spread more easily. The other definition here is a mutagen, and a mutagen is defined as a chemical or physical agent that interacts with DNA and can cause a mutation. So what are some examples of mutagens that you can think of? Some of these agents that cause mutations. Do we know any? What might be something that could cause a mutation? Okay, that's true. Crossing over is a process that can cause a mutation very early on. And that's a good point as well for another reason that'll come up later, Abigail. So thank you for mentioning that. What else? What are some other mutagens we can think of? Maybe we don't know any others. Okay. So here are some common mutagens, and if you didn't know these, then it would be a good idea to write down some of the examples. Radiation is a type of physical mutagen. UV sunlight or UV light is a type of radiation that can cause mutations in your skin cells. X-rays are a mutagen. In fact, if you've ever gotten an X-ray, they probably asked you to put on a lead vest so that your internal vital organs would not be damaged by the X-rays, especially if it was just an X-ray on your wrist or on your ankle. There are chemical agents as well. Carcinogens. A carcinogen is a cancer-causing agent, such as the nicotine in cigarettes. Processed foods and preservatives that are used to help the food last longer. So if you've ever wondered why you're able to buy watermelons in February when watermelons do not grow over the winter time, it's because of these preservatives. Or if you've ever wondered why apples look so perfect in the store, it's because they have preservatives to make them shine brighter and to make them have more vibrant colors. Cosmetics and cleaning products. So cosmetics, makeup, some types of makeup can also cause mutations. In fact, a long time ago, people would literally rub mercury on their skin to make it more pale. And that is extremely dangerous. So we've gone away from that, but even today, still some cosmetic products are not really that good for your skin. And then infectious agents, viruses and bacteria. We'll talk more about these later on in the course. Important to note that mutations can only be inherited if they are found in gametes, and these are your sex cells. So a mutation that occurs in a somatic cell is not going to be passed on to your offspring. So this is why I appreciated Abigail bringing up crossing over, because that is a process that takes place during meiosis. So it affects the gametes. Cool. All right. This is a long definition. You may want to write it down, but you don't have to. I just want you to, I will be referring to it for the rest of this lesson. So I just want you to really pay attention to what it means. The reading frame. So the reading frame is the non-overlapping triplet groupings of nucleotides. We call them codons that are translated into consecutive amino acids starting from a specific point. This is a fancy way of saying that when you read an mRNA, you start from a specific point, and then you read in groups of threes without skipping any, but you also don't overlap. So we'll see exactly what this means. You do not overlap, and you read consecutively, so you do not skip any letters. Okay, so for example, let's say we had a sequence of fake DNA. Of course, we have some letters here that don't appear in real DNA, H, E, F, blah, 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 R. If we were to take that sequence and then start the reading frame at the very beginning, it would look like this. We're reading in groups of three nucleotides. We call those codons. And then we translate them into consecutive amino acids. We're not skipping any. So the reading frame here is the fat cat ate the rat. 3-letter words. Okay, let's take another look at this. So types of mutations. There is a point mutation. And a point mutation is a change, aka a substitution, in a single nucleotide pair. So this would just be like replacing one nucleotide, putting a C where there should be a T. These are called point mutations because they're only affecting one point in the sequence. You're substituting one nucleotide for another. A C where there should be a T, or where there should be a G, an A where there should be a C. Okay, so let's take a look at one of these. Did you need me to go back, Ashley? Okay, so there's our original sequence again. The fat cat ate the rat. Let's say we change the R to a B. So now the new reading frame is the fat cat ate the bat. Notice here that we only affected one of the codons. The fat cat ate the, all five of those are unaffected. It's only that last codon that has been changed in a point mutation. Okay. This is important because point mutations do not shift the reading frame. They don't shift it over. We're starting from the same place and we're going to finish in the same place. And so they might only affect one codon. In some cases, they don't even really have an effect, and we'll see that later. They may only affect one codon. Cool. So for example, in that original sequence, we had TAA, CTG, CAG, GT. We've got a point mutation taking place at the bottom. So that very first codon, is it affected? The first codon, has it been changed at all? No, it hasn't. It has not been changed. It was originally TAA, and then even after the mutation, it's still TAA. What about the next codon? Has it been affected? Yeah, it's been changed. We put a C where there should have been a T, so that's messed up. What about the next codon? Is that one affected? No. It was CAG before the mutation, and it's still CAG afterwards. So these point mutations are not negatively affecting all of the codons, just one. Okay, so now my question is, what mRNA and amino acid do you get from the original sequence, CTG? What mRNA do you get from CTG? And I can type. So if my DNA is CTG, what is my mRNA? So I want you to transcribe it. GAC. So when we're transcribing, C is transcribed to G, T is transcribed to A, G is transcribed to C. Now the question becomes, what is the amino acid that you get from this? From GAC, what amino acid do you get? I keep forgetting to do this. Give me 1 second. Okay, so I just sent a link to the chat that you all can go to. It should take you to a codon chart for those of you who are at home. And I think I just put it on our Canvas page, so it might take a while, like 10 minutes or so, but it should be available on Canvas for you all to access, and you can add it to your bookmarks as well. So hopefully someone has figured it out by now. Which amino acid do we get? Good. ASP, which is aspartate, also known as aspartic acid. Please don't be frozen. Hold on. My remote's not, oh, there it is. Yeah, so we get GAC and aspartic acid. What do we get from the mutated sequence, which is now no longer CTG, but instead is CCG? First of all, what mRNA do we get from that? We get GGC. And then what amino acid do we get from GGC? Anybody at home figured it out yet? Because I'm getting some answers here from the folks in the classroom. We've got the mRNA GGC. Good, there we go. So Dijon, let's see, you got arginine. Where's arginine? Okay. So maybe you, I don't know how you got to arginine. Hold on. Oh, yeah, just make sure you're careful about I don't whatever codon chart you're using. Arginine is relatively close to glycine on this chart. On the circular chart, they are not all that close. But just make sure that you are, if you're using the circular chart, start in the middle of the cell and then move outwards. But yeah, we're getting the correct answer in the chat now. So GGC is the mRNA codon, and that gives us the amino acid proline. Is that right? No, glycine. If I made that mistake. Glycine, I'm sorry. But in any case, we see that the mutated amino acid is different, and this can greatly affect the resulting protein, especially if the amino acid is really, really different than what it should be. Amino acids have different properties. Some of them hate water. They're really, really hydrophobic. Some of them are, in love with water. They're really, really hydrophilic. And then there's a spectrum. So some of them kind of don't like water and some of them kind of do. So let's say we've got a place in our protein where there should be an amino acid that hates water, and we replace that amino acid with one that loves water. That's going to be a big problem, and it can lead to the protein being dysfunctional. But if we've got an amino acid that hates water and we replace it with an amino acid that kind of doesn't like water, the protein might still be able to function with that. Okay, so the properties of the amino acids are important. Okay, so again, nothing to really write down here, but oh no, this one is different. So I'm sorry, you should write this down. Frame shift mutations are changes in a nucleotide sequence that shift the entire reading frame. So if we start from the same point, we're not going to finish where we should. It shifts the entire reading frame over. Okay. So frame shift mutations can be either insertions or deletions of nucleotide bases. So we're not just substituting. We're literally going to put a new one in, or we're going to just delete one and take it out. And so this shifts the entire reading frame. And we can see an example of an insertion there at the bottom. It can be insertions or deletions. Everything after the point of insertion or deletion is altered. Everything that comes after that point of insertion or deletion is altered. Okay. So again, let's return to my sentence, my original sequence here. The fat cat ate the rat. Let's say we insert an X after the very first E. So after that E in the word the, that's going to be a frame shift insertion mutation. So here's what it would look like. So now we're starting our reading frame still at the beginning, so still in the same place. But instead of the fat cat ate the rat, we now end up with the XFA tika tat era. So the words don't make sense anymore. We've totally messed up the entire reading frame. All of the codons after the point of insertion are now messed up. Let's take a look at another one. This time we're going to do a frame shift deletion. So we're going to delete the F after the first E. We're deleting that F right there. So again, my very first codon is the, and it's unchanged. It remains unchanged. But everything after that is messed up. So with these frame shift mutations, we've got to think about everything coming after. Everything that's quote unquote downstream is going to be messed up. Now let's say we delete the R after the very last E. So now we're almost at the end of the sequence where we have the deletion. We're deleting that R right there. The new reading frame now is the fat cat ate the at. So for the most part, well, I'm going to let you guys draw the conclusion here. What do you notice about the difference between these two frame shift mutations? What's different about these two?
One is at the beginning and then one is at the end.
Good, Lisa. So one of them has a deletion at the very beginning, and one has a deletion at the very end. So what's the result of that? If the deletion is at the beginning, what happens versus if it's at the end?
The whole thing messes up.
Yes.
Looks completely different, while the other one looks the same, but there's no R.
Good, good. Right. So JT and Janelle was talking as well. Both of you said essentially the same thing. The earlier it is, the more damaged the sequence is going to be. If the mutation happens in the very beginning, it's probably going to ruin the protein totally. If it happens at the end, you might only have that very last amino acid that's messed up, and you might be able to deal with that. So the earlier the frame shift mutation happens, the more messed up the overall sequence will be. On the left, we've got the mutation happening on the fourth nucleotide. On the left, we've got it happening on like the 14th nucleotide, 13th. Okay. So let's take a look at this. Let's do some actual practice instead of with my fake sequences. So I've got a DNA sequence there. It's called the wild type. The wild type would just be kind of like the normal sequence. It's a fancy way of saying the normal sequence, which you would expect to actually see in the wild if everything was going correctly. So we've got TAC, TTC, AAA, CCG, ATT. Help me transcribe this first. What should be my very first mRNA nucleotide? A, then U, G, A, G,
G, U, G, G, C, U, A.
Good. So if you're still confused about this, that's okay. We're going to practice with it more, but please just make sure you're asking questions. If you're like, I literally don't understand this sequence at all, no big deal. I'm happy to help. It can be confusing. Really, it's just a matter of, taking the T, changing it to an A, taking the A, changing it to a U, taking the C, changing it to the G. We only have one G in the DNA sequence. It's all the way at the end, but you take that G and you transcribe it to a C. Okay, now let's do the real work. Let's translate this mRNA. What's the very first amino acid I get? And I kind of gave you guys a hint about this if you were paying attention a couple of days ago. Which one did I say we typically start with? Yeah, sorry, Melissa, I didn't see. Methionine, MET. And thank you for that as well. Oh, sorry. I messed up the order of my transitions here. But let's say we had a single nucleotide pair substitution. So this would just be a point mutation. We're just changing one nucleotide. So there's a T there. I'm going to change that. I'm sorry, there was an A. I changed it to a T. So in the wild type DNA, there was an A. And in the mutation, it became a T. So that messes up my mRNA when I go through this process of transcription. Everything else is still good, though, as we can see. So now I go back to translating. That very first amino acid, of course, should be methionine. Then what comes after that? What's the next amino acid? Make sure you're not overlapping. We don't want to start with G. We want to start with that next A. AAG is the codon. Lysine, yeah, LYS. Then what? What is it? So not in the mutated one, but in the wild type. Okay, thank you. I'm sorry, I'm not looking in the chat. So yes, it's phenylalanine. Phenylalanine. That's a weird word to say. Then the next one, GGC. Sayana, do you think you can do this? You're confused? Okay. So you've got this in front of you. Do you think you have a preference of which one you like to use? The circular one? Okay, so the next codon that we have is GGC, and a codon is just a group of 3 nucleotides. So if we kind of look at this chart as kind of like a what's it called? A quartile or whatever, are we looking at the top right, the top left, the bottom right, or the bottom left? We're looking at top left, good, because, and how do you know that? Good, and G is our very first nucleotide here. Then what? What do you do after that? The third letter is the C. Okay. So we know we're looking in the top left, so that already limits us to those 5, amino acids: glycine, glutamic acid, aspartic acid, alanine, and valine. So you found that G was the first, you told me G was the second, and C was the third. So what is the amino acid? Good, glycine. Okay, and then that very last codon is UAA. What does that give us? Good. That's the stop. That's one of our 3 stop codons, which tells the cell, or it tells the ribosome, translation is over. The protein has been made. You can stop what you're doing now. Okay. But we have a mutation on the bottom. The cool thing about these point mutations is that they do not affect every codon. They only affect the codon where the mutation takes place. So methionine, we still had that amino acid. We still had the next amino acid as well, lysine. But now we've got a mutation. So instead of UUU, we've got UUA. So what does that give us? Good, I'm getting answers in the chat. Sorry, I'm not looking at the chat today. Yes, we've got leucine, LEU. Good, UUA gives us LEU. And then everything after that is still correct. This specific type of point mutation is called a missense mutation. You don't need to write this down. But it's called missense because only one amino acid is changed. That could have an effect. It could be a really, really deleterious effect or detrimental effect. But it could also have an effect that's too small to really do much. Okay, let's do one more. So we've got the same wild type, nothing changed there. But now I did another substitution. I changed the G to an A. So my mRNA is going to be the same, except instead of having a C there, now I've got a U. So everything before that mutation is good, and everything after that mutation is good as well. I've got those same 3 amino acids, but now my codon should have been GGC, which would have given me glycine, but now it's GGU. So what amino acid do I get from that? Yeah, I'm still getting the same thing. I still get glycine. Even though that mutation is there, it doesn't change the amino acid. So this type of mutation is called a silent mutation because even though there was a mistake, it did not change the amino acid sequence at all. Why do you all think that happens? Why do you think that is? Or why is that a good thing, I should say? I know you really got to put your thinking caps on for this one. But why is it a good thing that even though we made a mistake in our process, it didn't ultimately change the protein that we made? Why is that a good thing? Any thoughts about that? Why is it a good thing? What'd you say? Yeah, so, okay, let me see if maybe I'm not asking the question right. How does that benefit? Well, okay, you're saying the right thing, Janelle. So I apologize. I guess I didn't frame this the right way. But what I'm getting at is that this provides an evolutionary benefit because, of course, cells are not going to be perfect all the time. So an analogy I can think of is if you have ever had a job and worked as a cashier. Let's say that you're working the register and your boss tells you, if at the end of the day this count is off by 1 penny, you're done, you're fired. That puts a lot more pressure on you to be perfect in every single transaction. But if at the end of the day your boss is like, we were off by $5, but that's cool, no big deal, then that's not really going to, you don't have as much pressure to be perfect. The cells work the same way. They know that they're not going to be perfect. They're really good. They really are quite efficient. In fact, they make a mistake once every trillion letters. So it is very rare that a mistake is made. Mutations don't happen very commonly, but they do happen. And when they do, we want to make sure that it doesn't totally disrupt the cell or the protein that is made. Another way that this protection is provided, if you look at these charts, these codon charts, the amino acids that are closer to one another are more similar to one another. And what that means is that if I, for example, have UUU, and that gives me phenylalanine, but then I have a mutation that makes me UUA, and that gives me leucine, that's not really a big deal because those two amino acids are pretty similar in terms of their chemical properties. So the cell, this is a factor of evolution, because the cell has come to terms with the fact that things are going to be imperfect. So when mistakes happen, we want to limit the end result. We don't want the end result to totally kill off the organism. All right. Last slide. Mutations, and we'll talk about this a lot more in our unit on evolution. Mutations can be harmful, they can be beneficial, or they can be neutral. Mutations have given us all of the diversity of life that we've seen on our planet. Mutations have given humans different skin colors. But if we go back millions of years, mutations were what allowed life to go from single-celled organisms to multicellular organisms. Mutations are what allowed life to go from living only in the water to life living on land. Mutations are what allowed organisms to go from being herbivores to being carnivores.
Once again, please complete the PD dash dark time survey. We'll get in the third one.
All right. What was I saying? Oh, okay. And then so some mutations are actually beneficial. So for example, we'll talk more about this. People who have sickle cell disease are also immune to malaria. Sickle cell disease is a disease that literally makes your red blood cells shaped like a sickle. It looks like a green bean instead of being shaped like a bowl. And the effect of that is that those sickle cells do not carry oxygen very well. This can be extremely, extremely dangerous, and it can also be quite painful because your extremities are not getting enough oxygen. So you can actually start to feel some of that tissue dying, and it can be really painful. But at the same time, people who have that sickle cell disease are resistant to malaria, which is another disease that's typically carried by mosquitoes, and leads to severe dehydration, vomiting, nausea, diarrhea, and it can also be deadly. So this is kind of a trade-off that nature discovered. We will be resistant to this viral disease that is being carried by mosquitoes, but in exchange, we have to, get this other disease. So what we find is that people who have grown up in, or not grown up, but people who have ancestors from areas of the world where there were once a lot of mosquitoes are more likely to today have sickle cell disease. So this means that people who are descended from Western Africa are more likely to have sickle cell disease, but also people who are descended from Central America are also more likely to have sickle cell disease because their ancestors were fighting off these mosquitoes, and so they did this genetic trade-off of sorts. So it's an interesting case study. We'll talk more about that, but it's caused by literally 1 point mutation. Where there should be an A, there's a T, and this leads to this, these two diseases, or leads to the one disease that prevents the other. So it's really quite remarkable. All right, we have how much time? Oh, we only have 4 minutes. My apologies. I thought we had way more time than that. There is an exit ticket. There's also an assignment. The assignment, I think, will be pretty quick for you all. Excuse me. It's going to be a review of something that we talked about at the very beginning of this unit: DNA structure, function, and DNA replication. So I just called it the DNA overview assignment. Tomorrow's asynchronous assignment will focus on transcription and translation, and we'll keep reviewing in preparation for Tuesday's midterm. So I might change the due date for the assignments because your midterm is not until Tuesday. So I'll probably make the midterm, or I'll make the assignments due on Monday. So you all will have the weekend to work on them. Are there any questions?
Scott, do you have any like work like paper that can make me understand the codes a little bit more? Like how they translate it to R in RNA to amino acids. Like, do you have any that can make me more understanding that?
I don't currently have one, but I can definitely put together something for you. So I'll have something for you tomorrow.
Okay, thank you.
You're welcome.